Hello students, in this lecture we are going to do the morphology of the seed. Before we begin on the morphology of seed, I would like you to complete 2 lectures whose links I am providing in the description below. The first lecture is on the structure of the ovule and the second lecture is on post-fertilization developments. Once you complete these 2 lectures, come back to this lecture. So let's begin. The seed. The seed is formed from the ovule. Post-fertilization. Ovule post-fertilization forms the seed. Integuments of the ovule post-fertilization forms the seed coat. The zygote post-fertilization forms the embryo, and the primary endosperm nucleus, which is formed as a result of the double fertilization. You know that there are 2 fertilizations that take place inside the ovule. First is the fertilization of the egg cell with the first male gamete resulting in the formation of the zygote. The second fertilization of the second male gamete occurs with the secondary nucleus giving rise to the primary endosperm nucleus, which post-fertilization becomes the endosperm. So a typical seed will have 3 parts in it. First part will be the seed coat, second part will be the embryo, and the third part is going to be the endosperm. The embryo is divided into 2 parts. Embryonal axis. The embryonal axis comprises of the longitudinal plant and the cotyledons. You know that monocots have only 1 cotyledon and dicots have 2 cotyledons. And then there will be the endosperm. Dicots are known to not have an endosperm and monocots have endosperm present. In case of dicot seeds, the embryo consumes the endosperm and becomes fat itself. And in case of monocot seeds, the endosperm is left as it is, so it will remain distinct. So right now we are doing the structure of a dicotyledonous seed, for which we are taking the example of gram seed. Chana. You can ask your mother to make chole tonight. Observe one gram seed very closely. So this is the gram seed. This is closed and this is open. Let's talk about each one of those aspects for the dicotyledonous seed. Let us start with the seed coat. Seed coat is the outermost covering of the seed. You've seen it in case of gram, right? The covering just comes off very nicely. It is formed from integuments of the ovule. If you remember, there were 2 integuments of the ovule. A typical angiospermic ovule is bitegmic. There is an outer integument and then there is an inner integument as well. The outer integument will give rise to the outer cover of the seed coat, which is called as testa, and the inner integument of the seed will give rise to this layer called as tegmen. Both testa and tegmen together will form the seed coat. Testa is more hard and tegmen will be more membranous kind of in nature. You may not be able to see it very distinctly in case of the seed coat, but it is there. Onto that seed coat you will find white kind of a scar, not very distinctly seen on a gram seed, but you can see it very distinctly on other seeds like a bean seed. A white color scar mark is present. Also a chiku seed, a long prominent scar is present on it. That is the place where this seed was attached to the fruit. The ovule is attached to the ovary via a funiculus. Okay, funiculus is then connected to the placenta. So when the ovule comes off, it leaves a mark on the seed coat. That mark on the seed coat is known as a hilum. Hilum can be defined as a scar on seed coat, which is a point of attachment of ovule with funiculus, clearly seen as a white scar in chiku seed. Right above the hilum, there will be a small pore. This pore is the micropyle, the same micropyle which was also present in case of the ovule. What is the purpose of this micropyle? It is the point of entry of water and gases. Micropyle, an opening, a pore opening above hilum, allows entry of water and air in the seed, after which only the seed will germinate. So that is about the first aspect of the seed, that is the seed coat. Next we move on to the embryo of the dicot. So if we remove the seed coat and if we gently open this seed, you will find that there will be 2 such big structures, which will be the 2 cotyledons. The 2 cotyledons, because of which this is a dicotyledonous seed, which is the case for all the dicotyledonous seeds. They all will open in the middle into 2 such kind of halves, which is not the case with monocot seeds like in maize. Does not open in that manner. But pea, bean, gram, moong, dal, all these seeds open in the middle. So these are the 2 cotyledons, and this is the embryonal axis. So now let's talk about this embryo aspect. Embryo has two parts. Let's talk about the embryonal axis, the first part. So if I call this as the embryonal axis, it is divided into certain regions here. 5 distinct regions can be marked. This is the first part. This is known as the plumule. Plumule can be defined as the embryonic shoot. Next, what about this part below? This part is the radicle, which is the embryonic root. These things are the nodes. It is from the region of nodes that these two cotyledons have emerged. Did you know that cotyledons are embryonic leaves? The leaves arise from nodes, isn't it? Over here also, the two cotyledons, the embryonic leaves, have arisen from the nodes itself. Make a mention here, embryonic leaves. Then this region between the node and the plumule, this part is known as the epicotyl. This region between the node and the radicle is known as the hypocotyl. Now these terms, epicotyl and hypocotyl, are not there for you in this chapter, but you will find them in NCERT subsequently. So you better know what is epicotyl and hypocotyl. So that comprises of your embryonal axis. Moving on, cotyledons. In dicot seeds, there will be two cotyledons. Two large fleshy cotyledons. Why are they so fleshy and large? They are so large and fleshy because these cotyledons have entirely consumed the endosperm of the seed and have become large and fleshy themselves. The entire endospermic food is now inside them. Since they consume the entire endosperm, which is also the reason why dicot seeds are not endospermic seeds. Third part. So we are on the third part, endosperm. Dicot seeds are, fill in the blank, are non-endospermous, also called as Exalbuminous. Also called as non-endospermic. Dicot seeds are non-endospermous or Exalbuminous seeds. But there are a few exceptions. One exception is Castor seed is a dicot but endospermous seed. This is an important example. It is frequently asked. A castor seed is a dicot seed but it is endospermic seed. So this is all we have from the structure of the dicotyledonous seed. Most important thing to know in this is the dicot seed will have two cotyledons which will be fleshy and they will be non-endospermic. In the next lecture we will do the structure of the monocotyledonous seed.
In the name of Lord Mahadev, please subscribe.